More potent vaccines utilizing RNA are being developed by engineers. For respiratory illnesses like COVID-19, a possible solution is intranasal vaccines, which the novel method could provide. At MIT, a group of researchers are improving RNA vaccines for COVID-19. The study proved that these modified vaccines can produce a better immune response in mice at a reduced dosage. This design adaptation could lead to coronavirus vaccines that are even more effective than current ones. In the quest to stimulate the immune responses of recipients of vaccines, adjuvants are almost always used. But have you ever heard of an RNA vaccine incorporating this? The leaders of the Massachusetts Institute of Technology delved into this frontier and created nanoparticles and COVID-19 antigen-combined engineering that would do the job of an adjuvant. Such a breakthrough means that a separate adjuvant isn't necessary anymore for boosted immune responses, potentially leading to longer-lasting immunity, reducing dosage requirements and cutting costs. This RNA vaccine is primed for use in humans. To boot, researchers discovered the vaccine incited a potent immune response when administered intranasally, significantly stronger than the traditional intramuscular method. Daniel Anderson, the senior author of the study and a professor in MIT's Department of Chemical Engineering, as well as a member of MIT's Koch Institute for Integrative Cancer Research and Institute for Medical Engineering and Science, IMES, proposed the intriguing notion that intranasal vaccination could potentially eradicate COVID at the mucous membrane, preventing its entrance into the body. Moreover, he adds, intranasal vaccines possess the advantage of not needing an injection, which might facilitate their administration to a larger population. Incorporating similar immune-stimulating properties, the effectiveness of other RNA vaccines being developed for cancer and other illnesses could potentially be enhanced, according to the researchers. Appearing in Nature Biomedical Engineering is a new study co-authored by University of Toronto Assistant Professor Bowen Lee, graduate student Alan Jiang, and previous Boston Children's Hospital Research Fellow and former MIT postdoc Idris Raji. Many others contributed to the work, including David H. Koch Institute Professor Robert Langer and a number of other MIT researchers. A lipid nanoparticle carrier is used to package RNA vaccines to shield them from disintegrating within the body and to assist them in entering cells. These vaccines consist of an RNA strand that generates a viral or bacterial protein known as an antigen, such as a portion of a COVID-19 virus's spike protein. The SARS-CoV-2 virus can provoke antibodies and T cells within the immune system by converting the transported RNA into proteins upon cellular arrival. These proteins may then be recognized by the immune system if the individual is ever infected with the virus. By engineering COVID-19 RNA vaccines to have immune stimulatory properties, the MIT team aimed to improve the already strong immune responses provoked by Moderna and Pfizer-BioNTech's original versions. The immune response was bolstered in this research through two distinct approaches. One of them was centered around C3d, a protein from the complement system branch of the immune system. This group of proteins battles infection, with C3d's task being to attach to antigens and enhance the body's antibody response towards them. As a molecular adjuvant for protein-based vaccines, like the DPT vaccine, C3d has been a subject of scrutiny by experts for quite some time. Jiang saw potential in utilizing C3d as an adjuvant in mRNA vaccine systems, especially with the success seen in COVID vaccines. This would be a fantastic opportunity, Jiang said. The mRNA was manipulated by the researchers to contain the antigen fused with the C3d protein, allowing for a simultaneous production of both components in cells that have been vaccinated. The lipid nanoparticles were altered by the researchers in the second phase of their plan. Not only do these lipids assist with RNA delivery, but they also stimulate the immune response intrinsically, all in an effort to improve the RNA vaccine. The researchers established a collection of 480 lipid nanoparticles with varying chemistries to determine the most effective lipids. These lipids all have the ability to ionize and become positively charged in acidic environments. Previously, COVID RNA vaccines contained ionizable lipids which aided nanoparticle, RNA self-assembly and targeted cells to receive the vaccine. Rather than aiming for perfection, the researchers at first weren't completely certain which chemical concoction would bring about the most effective immunostimulatory response with nanoparticles. We knew that nanoparticles could have this effect, but what was necessary to optimize it eluded us, explains Anderson. In response, a library was created for evaluation purposes, leading to the discovery of certain chemistries that showed promise in stimulating a strong response. The vaccine, a result of combining RNA-encoded C3d and the superior ionizable lipid, was tested on mice. Among mice tested, the vaccine utilizing adjuvant technology resulted in production of antibodies 10 times greater than the unadjuvanted COVID RNA vaccine. Furthermore, the vaccine produced a robust response within T cells, a crucial defense against the SARS-CoV-2 virus. Investigating the feasibility of administering the new RNA vaccine platform intranasally was a natural next step after seeing a synergistic boost in immune responses by engineering both the RNA and its delivery vehicles. The challenges presented by the mucociliary blanket barrier in the upper airways were taken into consideration during this process, according to Lee. This marks the first time such a boost has been demonstrated, he adds. An intranasal vaccine could potentially provide more comprehensive protection against infection by inducing an immune response in the mucosal tissues lining the nasal passages and lungs, according to researchers. They noted a comparable immune response in the mice when the vaccine was administered through the nose. If approved for human use, an intranasal vaccine option could be highly beneficial. The researchers share that self-adjuvanting vaccines are not only cost-effective, but also enable a stronger response with a lower dose, making them more accessible to individuals in developing nations. The lab of Anderson is looking into the potential of their self-adjuvanting platform to enhance the effectiveness of various RNA vaccines, such as those for cancer. Collaborating with healthcare companies, they have plans to assess the safety and efficacy of these innovative vaccine variants in larger animal models before moving on to human trials. Translate Bio and the National Institutes of Health. Each contributed funding to the research. To support our channel's growth and ensure broader awareness, kindly hit the like and subscribe buttons. This will help us reach more individuals and disseminate valuable information. Thank you in advance.